Okay, can somebody please confirm you can hear me?
Yeah.
Thank you. Today is April 25th, and we're supposed to be starting chapter 4. Can somebody inform me where we left off? I'm trying to find that now. Let's see if we finished chapter 3.
Before we start, sorry to interrupt you. Before we start the chapter, do you think we'd be able to talk about moving the quiz time to 12 to 12?
We can talk about that, yes.
Okay, well, I know there are some other classmates that don't really like the 8 to 8 because it interferes with their schedule. So we've been talking about if you would be willing to move it from 12 to 12.
Okay, like I said, if students want me to, I will make a post, what do you call that? A discussion where students can comment and then vote on what they would like.
Okay.
So there's been a request by some students to change the quiz time from 8 p.m. on Tuesday till 8 p.m. on Wednesday to make it, let's say, 12:01 on Wednesday and end at midnight Wednesday night. So you'll still have 24 hours to get the quiz done. It's just a different 24 hours. Now, I had it initially at midnight, and students who were working, meaning midnight to midnight, students who were working found that that didn't fit their schedule very well. And when they came home, they were kind of tired and they didn't really want to take the quiz after they came home. And I don't know what their working hours are, but let's say it's regular, something like 9 to 5. If you are working and you wait until after work and then you come home and take the exam, you will be tired after working. So understand that. It obviously depends on what your work schedule is. Somebody who's working like in the middle of the night isn't going to like, well, probably any of the schedules I'm going to give you. The 8 to 8 p.m. I said was set up for people who were working. The midnight to midnight works well for students who are regular students and are not working, especially after class.
Okay, well, like you said, you're gonna set up the discussion post, so.
Yeah, I will set up a discussion post.
And if it
There's been a
doesn't
few
work
people.
if it doesn't work for most of the class, then I'm sure we'll be able to find out there.
Yeah, you'll find out. I'm not sure what I'll do if a few people object to moving from midnight to midnight, because I've got it set up 8 p.m. to 8 p.m. But if everyone wants it midnight to midnight, I will switch it that way for sure. Okay, if the majority want it midnight to midnight, I'll have to consider what to do. But like I said, if there's a few that object, I will have to decide what I want to do, because I have set it up 8 p.m. to 8 p.m. If nobody objects, then I'll switch it to 12 to 12 for sure. Okay.
Dr. Volkan, I had a question. And would this go into affect this quiz or
No, it
next
won't go
quiz?
into affect this quiz because I'm going to have it set up a discussion board, and obviously some students aren't on Zoom right now, and they might not see this class until, I don't know, Friday or Saturday. So it will be definitely, when's the next quiz? It's not going to be this week for sure. This is April 25th. All right, this week we do not have a quiz. Well, yes, we do. Sorry, I'm not seeing too well. It will not be affecting this week's quiz. So quiz 2 will be held at the regular time. And something like Saturday, I will then read the discussion and then make my decision. So quiz 3 may be changed, although that's only a few days. I'm not sure when I'll change it, assuming that we change it, but quiz 2 will definitely be at the regular time because that is this Wednesday, and I'm not going to end the discussion until Saturday at 11:59 p.m. Okay. And that's only going to make a few days for quiz 3. So quiz 3 will probably be done at the regular time, meaning 8 p.m. to 8 p.m. Because I would like to make this announcement and then have a week before we switch over. And then the next week, there is no quiz, so that will work well. All right, any other questions about that or any other topic? Okay, like I said, on the schedule, we're supposed to be starting chapter 4. There is a quiz, quiz 2, this week, which will be from 8 p.m. to 8 p.m. So Tuesday at 8 p.m. to 8 p.m. Wednesday. Chapter 2 covers labs 3 and 4, chapters 3, and the end of chapter 2. There is going to be a lab today, lab 5, so do show up at the lab time. Now I've got a question for you. Where did we leave off? Did we finish chapter 3?
We were talking about the gram stain procedure.
So we're on chapter 3 right here?
Yeah, I
can't see
believe
that.
so.
Let me show my screen here. Come on. Oh, shoot. Just a minute here. I'm trying to get my screen so I can share it. Now get that out of the way. Oh, there we go. So I've got a note saying that we stopped here on the gram stain. So we did cover the gram stain procedure in basics. So we're now going to talk in great detail about the gram stains. Everybody agree that's where we were?
Yes.
Okay. All right, now let me stop sharing my screen and I'll talk a little bit about the grading. There we go. So I've done some grading and I'm trying to get, let's close that down and that down. I'm trying to get the last lab we had done graded, but I was not able to get to it today. I've got about half the students graded, maybe a little less than half. Where's the grade book? Trying to figure out what lab that was. It's the last lab you turned in. It's grading the first time. I'm not seeing too well today. I'm not sure why that is. Maybe I should clean my glasses. Now I can't see it all, but yeah, so lab 4 is the one I'm working on. I've only got close to half the students graded, a little less maybe. And I have graded previously lab 3 and lab 2 and lab 1 the first time. Lab 1 has been graded the second time, except for a few students. I'm not sure why there's a few students that have not been graded the second time, but most students have been graded the 2 times on lab 1. I also got graded worksheet for lecture 2 and extra credit study group 2. I think I did one of the extra credit quizzes, and if you look at your score, it'll look like it's not graded, but if you click on it, it'll have a score in it. It's practice quiz 00A. It's not showing correctly in the grade book, and I'm not sure why. It is graded. And then practice quiz 00B is also graded. On practice quiz 00B, you got 0.1 point for each correct answer. And that one is showing correctly on the grade in the grade book. Let me remind one student, I sent them an email that the grade book is doing something funny with at least some of their grades. And it wasn't, when I put in a grade in the grade book, it was showing the incorrect score. And I'm not sure why that is the case. So I went in and changed that student score manually in the grade book. And I'm not sure what's going on. I'm guessing it's a bug in Canvas. Unfortunately, it's only happening with one student, where I put in the grade in the assignment, and then it's not showing the correct grade in the grade book. So that student, you're going to have to watch your grades very carefully this term, and I'm going to try and watch it carefully as well. Maybe this will only happen one time, but we'll have to find out. All right, any question about the grading?
I have a question about the slides.
Uh-huh.
Is there any way that you could post it prior to the lecture so we could follow along and take notes with you?
I've already answered that, and that is no. I want you to take your own notes. I don't want you to use my notes. The reason being is that students learn best from taking their own notes. Okay?
Okay.
Now, if you want my notes, you can go in and get the study guide.
Okay.
Okay?
I think, okay, that's fine.
It's actually more work for me, but when it benefits my students, I will work that way. And there's no question. Let me make chapter 2 available to you. Whenever I don't have the notes, the slides available, after we're finished with them, you should let me know, by the way. Did we finish chapter 3? I don't think so. I think that's where we are. Yep, all right. Okay, any other questions? Let me share my screen again. Actually, I better get off the grade book in case I happen to go. There, don't want to show the grades. All right, let me share my screen now. All right. So I think we talked about this slide. Initially, when you stain with crystal violet, all the cells stain purple, both the gram-negative and the gram-positive. And some of you must not be following the lecture very closely, because I clearly stated that. And in the homework assignment, you got this wrong. And then we stain with iodine, and the cells just are more intensely stained. It's a little darker purple, more purple. It's blue purple with just the crystal violet. And then we do the decolorizing step, and the gram-negative cells are clear. The gram-positive cells, if the gram stain is done correctly, meaning the decolorizing step is done correctly, the gram-positive cells remain purple, but the gram-negative are clear. And then we stain with safranin. It is at this point that the cells become pink, and that's because safranin, the counter stain, is pinkish or reddish in color, and the gram-positive cells remain purple. All right, so before we do the gram stain, and actually before we do any stain, generally we make a bacterial smear. And this is another question that a few students got wrong. And that is, when you're doing the bacterial smear, you are not doing any staining. You're not doing any steps of the staining. A bacterial smear is making the cells on the slide, and that is a procedure we do before we stain the cells. You then air dry and heat fix the cells on the slide. That's part of the bacterial smear. And that's steps we do also before we do any staining. We then do the staining procedures. When we're staining with gram stain, we flood the slide with crystal violet, meaning we put crystal violet all over the slide, and we leave it on for 60 seconds or a minute. After that time, you then dump the crystal violet off and then wash the slide with cold water. You gently wash to reduce the chance of knocking the cells off, although the cells are air dried and heat fixed, so they generally will stay on the slide. Crystal violet sticks to the peptidoglycan in the bacterial cell wall. Crystal violet is considered the primary stain since it's the first stain used, and it colors all of the cells purple, and it's a blue-purple color. So both the gram-positive and the gram-negative stain purple. We then apply the iodine. Obviously, you dump off the water. And iodine is not a stain, it is a mordant. It increases the affinity of crystal violet for the specimen. The iodine actually forms a complex with the crystal violet, making a larger molecule. You apply the iodine for 60 seconds or a minute. You then dump it off and then wash the slide once again with cold water. The color of all cells, both gram-positive and gram-negative, is purple, although this is a more purple color than the crystal violet alone. It's also a little more intensely staining. Although to the human eye, you can't really tell the intensity. We then do the decolorizing step. And obviously after the iodine, did I state that there? Yeah, you wash with cold water, do have it there. You then do the decolorizing step, and the decolorizing agents can be different agents. The most commonly used is an acetone ethanol mixture, but you could have an acid ethanol mixture or just an ethanol mixture, which isn't a mixture, it's just ethanol. Sorry. This decolorizing agent washes the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells. This is when you're applying the decolorizing agent for the correct amount of time. If you apply it for too long, you will wash the crystal violet iodine out of the gram-positive cells. And if you don't wash long enough, you will not wash all of the crystal violet iodine out of the gram-negative cells. Did I say that wrong in the first time? The first time, if you have the decolorizing agent on too long, you will wash the crystal violet iodine out of the gram-positive cells. I may have said that wrong the first time. This step is considered the differential step in the gram stain, because at this step, we see differences between the gram-positive and the gram-negative cells. You apply the acetone ethanol mixture for only about 5 seconds. And in reality, the best way to apply it is to watch while you're applying it. So I'm going to try and show you. Can you guys see this? I'm going to pretend this yellow thing is the slide and this pen is the eyedropper that you're applying the decolorizing agent. Can you guys see this?
Yes.
All right, great. So what you do is you apply the crystal violet iodine, and as soon as it stops washing, meaning the purple stops running, you stop applying the decolorizing agent. And that will be about 5 seconds. Okay, so you watch while you're doing it and you apply it. And initially the purple will run, and once the purple stops running, you stop applying the decolorizing agent. You then immediately wash the slide with cold water. You need to do the wash immediately because the slide will have minute quantities of the decolorizing agent on the slide, and that will pull out, meaning continue acting, will pull out the crystal violet iodine complex from the gram-positive cells. So you want to remove that by washing immediately. This is the only time where you need to wash the cells immediately. At all the other times, you can do it when it's convenient. Okay. So at this time, the gram-positive cells should stain purple if the decolorizing agent was applied correctly. And then the gram-negative cells should be colorless, assuming that the decolorizing step was done appropriately. All right, you then dump off the water and apply the safranin. Safranin is a pink or reddish dye. It does get in all of the bacteria and it does stain all of the bacteria. Safranin is a counter stain because it has a different color to the primary stain, meaning a different color than the crystal violet. You apply the counter stain for 1 to 2 minutes. And in my procedure, I always use 1 minute because at Clark College, I found that the concentration of the safranin is strong enough that 1 minute is all you need to do. You then dump off the safranin and wash the slide with cold water. The gram-positive cells stain purple. The gram-negative cells stain pinkish. You then blot dry the slide with bibulus paper. And let me see if I can kind of show that. Of course, it'd be better if I had something that's a different color than what I'm going to use. I don't have bibulus paper with me, but you get some paper. Oh, this would be good enough. I don't know if you can see this. I'll try and hold it up. Put the slide in the bibulus paper, and it's best to actually rip the bibulus paper off, which I don't want to do that here. So let me get another piece of paper. And then fold the bibulus paper over the slide, and then just blot it. I don't know if you can see this, but you fold it like that, or like that, whatever way, and then blot it. You do not rub the slide against the paper. That will rub off the cells. So you just blot the paper by just pressing down. That will remove most of the water from the slide, and it will remove enough water from the slide that you can then take the slide and view it under the microscope. And you may do this even if you use oil, meaning enough of the water is removed that you can use oil on the slide. Any question about any of that? All right, so the gram-positive cells are purple, The gram-negative cells are pink. Why? What is the basis for the differential staining? It has to do with the thickness of the cell wall. There are differences in the cell wall between gram-negative and gram-positive cells. Gram-positive cells have a thick peptidoglycan layer. We'll talk about what peptidoglycan is in our next lesson. It's a disaccharide and has amino acids in it. And there's many, many layers. I'm talking about over 100 layers of peptidoglycan in the gram-positive cell. The gram-negative bacteria do have a layer of peptidoglycan, but it is thin. It does have many layers, but it's on the neighborhood of about 20 layers. It also contains lipopolysaccharide on the outer portion of the cell wall. And we'll show that here. Let me blow this slide up. I can get to it. There we go. So the gram-positive cell shown here, there's the cell membrane in yellow, the cell wall in purple. It's thick. When we look at this, there's many layers. Oops, didn't mean to do that. It's only 3 shown here, but it's actually, like I said, closer to 100 or many more, meaning maybe even 1,000 layers. But it's depending on the cell, but it's at least 100 layers, okay, for the gram-positive cells. The gram-negative cell has the cell wall with the cell membrane here, then a thin layer of peptidoglycan, thin compared to the gram-positive cells, and then a lipid layer outside the cell wall. And there we're seeing it there. Let me see if I can blow this up a little. The cell membrane, the cell wall, it's only showing you one layer. Realize it's more than one layer. It is at least several layers, and in reality, it's closer to 20 layers of peptidoglycan, okay? And outside of the peptidoglycan, or on the outside of the peptidoglycan, is a lipid layer, and that we call the outer membrane of the cell wall. We'll talk more about the makeup of that in our next lesson. Any questions about any of that? Oops. All right, so the crystalviolet iodine complex forms in all cells. Crystalviolet iodine is larger than crystalviolet or iodine alone. In the gram-positive cells, the crystalviolet iodine cannot be washed out of an intact thick peptidoglycan layer as long as the decolorizing step is done appropriately. The cells therefore remain purple. In the gram-negative cells, the crystalviolet iodine complex is washed out of the thinner peptidoglycan layer. And then the lipopolysaccharide layer is disrupted by the alcohol wash. So the crystalviolet iodine complex washes out and the cells become colorless. Here we're looking at a gram stain. There's gram-positive rods, gram-positive cocci, gram-negative, where's a rod? Right there. Gram-negative spirilla right here and right there. There's a coccus. I think that's gram-positive, but I can't really see what color that is. It's not blowing up. Yeah, this gram-positive coccus. Oops, didn't mean to go that small. All right. Now, there are some problems with the gram stain. I'm not seeing my note here. Like, what do you do when you have this on the slide, where these cells look kind of pinkish, and then these cells right here are definitely dark purple, and then those cells right there are darker purple than these cells there. which I guess that's not very pinkish right there, but it's slightly sort of light purple is what it is. So when you have something like this, these are bad regions of the slide. And when you're reading a slide, you have to look at the good regions. Let's shrink that down a little bit. The good regions of the slide, where the cells are distinct. They're not clumped together, like right there, they're clumped together. Where there are not crystal violet crystals, actually that right there, that's not a cell or garbage, or right there or there. What that is a crystal of crystal violet. Crystal violet, when you make the solution, especially if it's been sitting for a while, tends to form crystals, and then those crystals go to the bottom of the, fall to the bottom of the bottle or eyedropper. What do you call that? Sure, stick the eyedropper in, whatever. And then you can put that on the slide. And usually around these crystals, these cells will stain darker purple. And so if you have gram negative, and that's not really gram negative, then these cells here will appear gram positive. So you have to ignore the region of the bad region of the slide and then make your gram stain in the good regions of a slide. Any question about that? The gram stain has a few other problems, meaning most consistent results are obtained when performed on young growing bacteria. If you don't have young growing bacteria, and I'm thinking about bacteria that are less than 2 days old, you should not do the gram stain because you may not get correct results if the cells are older than 2 days old. Some bacteria don't stain well, so they stain poorly, if at all, with the gram stain. So the results are not always as clear-cut as the theory suggests. And this may become evident when you're working on your unknown project. Sometimes you'll be pulling your hair out saying, what's going on? So the main point is if you're doing the gram stain, make sure you have cells that are less than or equal to 2 days old. If they're older than that, you should grow up fresh cells before you attempt to do the gram stain. The gram positive and gram negative cells have more differences than just their makeup of their cell walls. They also differ in how they respond to antibiotics. Like penicillin G is easily killed or easily kills gram-positive cells, but penicillin G does not work on gram-negative cells. Gram-negative bacteria are resistant to penicillin G, as well as some cephalosporin and some members of that family, which is escaping me. What the family is cephalosporin? I forget. And then some detergents don't work well on a gram-negative cell, and they tend to work well on gram-positive cells. What happens in the gram-negative cells prevent the penetration of these ingredients through the lipopolysaccharide layer, meaning the gram-negative cell has an outer membrane around their cell wall, and that prevents penicillin G, as well as cephalosporin, from getting inside the cell and then doing damage to the cells. And it does prevent some detergents also. All right, any questions about the gram stain? If not, let's talk about some of the special stains. You will not be examined on the special stains. I want to just talk about it because you may need to do at least the capsule stain, which is a special stain, staining for a capsule. You may need to do that for your unknown project. You use the capsule stain to determine if your cells produce a capsule, as shown here. This is a negative stain ink. It is a capsule stain, and India ink stains the background very darkly, meaning it's an acid stain, not acid fast, acid stain, and it is also a negative stain because it makes the background dark. It does lightly stain the cells, but it does not stain the capsule around the cell. Let me blow that up. So that cell is lightly stained by India ink. The capsule is not, and that will look like a clear zone around the cell, and that is from a capsule. All right, any questions about this chapter? If not, let's move on to chapter 4. I guess while I'm here, I can make the notes available. Chapter 4, functional anatomy of prokaryotic and eukaryotic cells. We will definitely talk about the anatomy of prokaryotic cells. We will only talk about the anatomy of eukaryotic cells if we have time. And since we're behind schedule, I doubt we'll have time. Okay, this slide is giving you the major goals and a rough outline. Do you know the terms of this chapter? The endosymbiotic theory is one of the terms, and I will talk about the endosymbiotic theory, even if we do not talk about other characteristics of eukaryotic cells. Okay? The endosymbiotic theory is a theory to explain essentially how eukaryotic cells became what they are today. We'll also talk about how gram-positive and gram-negative cell walls differ and be able to describe the importance of taxis in bacteria. So when we're looking at prokaryotes and eukaryotes, you should know that prokaryotes is a Greek word for pre-nucleus. Literally, it means pre-kernel, but we don't call it kernel, we call that a nucleus. And prokaryotes do not have a nucleus. Eukaryotes is a Greek word for true nucleus or true kernel. And that's the main difference, although there are other major differences between prokaryotes and eukaryotes. The other main difference between prokaryotes and eukaryotes are prokaryotes have no membrane-bound organelles. I tend to shorten that to just organelles, because if ever I use the word organelle, I am meaning a membrane-bound organelle. Okay, that's a little different than the way the book uses that term. They do use organelle for a membrane-bound organelle, but they also use organelle for what I call a cell structure, and that is a structure in the cell that does not have a membrane around it, such as the mitotic spindle fiber or the centriole. Those are cell structures to me, but your textbook author does call them organelles, and it's a non-membrane bound organelle. That's the other way where eukaryotes different. The membrane bound organelles there are membrane-bound organelles in these cells. And those are the two main differences between prokaryotes and eukaryotes. There are a few other differences, like prokaryotes, or at least bacteria, typically have one circular chromosome, where the chromosome is in a circle. The beginning place is the same place as the ending place. Eukaryotes mostly have several chromosomes or many chromosomes, and they're not circular. Like the X chromosome has a beginning place and an ending place. Of course, the linear chromosomes are in the nucleus, and the chromosome of a bacteria is not in a nucleus because there is no nucleus. There are some differences in the histones. Prokaryotes do not have histones on their DNA, and they do not have histone proteins in the cells, by the way. Histone protein, as far as I know, although it's made in the cytoplasm, it moves into the nucleus of a eukaryote, and that histone will then associate with the DNA. The DNA actually wraps around the histone about 2 times and then moves on to the next. The strand of DNA then moves to the next histone. But my point is that the DNA wraps around the histone. So that's another difference. Another difference, which I guess is a major difference, I forgot about this one, and that is in bacteria, we can find the molecule of peptidoglycan. So it's only found in bacteria. In the archaea, they mostly do not have peptidoglycan. Okay, so we're going to say the archaea do not have peptidoglycan, and the eukaryotes do not have peptidoglycan. Even if they have a cell wall, which most eukaryotes do have a cell wall, it's only animals and some of the protists which do not have a cell wall. And then another difference is the way the cells divide. Prokaryotes tend to divide by binary fission. We'll talk about that later, but in binary fission, one cell splits evenly into 2 cells. And eukaryotes divide by mitosis. Any question about any of that?
I had a question about the centrosomes. So the centrosomes are, what's the difference between the centrosomes and the centrioles?
A centriole is a portion of the centrosome. And exactly what the difference is, I don't remember, so let me look it up. I know the centrosome is found in animal cells, and it's important in laying down the mitotic spindle. Actually, in this definition, I don't see any difference. It's an organelle making the mitotic spindle. And let's go look at this picture here and says anything. Can anyone see that picture good enough? Because I didn't. Yep, don't want that. Let me see if I can blow this up and read it. Well, that didn't work well. Let's go picture. Oh, here we go. Can anyone read the slide? So the centromere is this region of a cell, and it does include the centriole. It does have a few like mitotic spindles around it in this region, but the main mitotic spindles go this way from one centriole to the other. So the centromere is just the region around the centriole. Okay, so those, you can't see it, but that right there is this right here. And then that one there is this one here. So the centrosome is just the region around the centriole. Okay? Hopefully I didn't shut down the thing. All right. Okay, so let's talk about the cell shape and arrangement of prokaryotes. We'll move on to the cell structures and then the movement of materials across membranes, which in prokaryotes is only the cell membrane. Prokaryote cell sizes and shapes. Their average size is, actually the average is about 1 to 2 micrometers in diameter. The length can be longer, but then the diameter there is close to 1 to 2 micrometers long. So they can be longer than they are in diameter. And then it really depends on what author you're getting. The smallest cell, prokaryote, is about 0.4 micrometers in diameter. And then the largest eukaryote cell is the, at least on the planet Earth at the moment, is the yolk, the egg yolk of a ostrich egg. Okay, so that's just an average sort of. The prokaryotes have 3 basic shapes. They have round, and we call those coccus. If there's more than one, we call it cocci. And there you can pronounce cocci 4 different ways. Let's see if I can remember them. Cocci, cocky, coxy, and coxi. Okay, all of those pronunciations are correct. I just go cockeye. We also have the main shape bacillus shown here, and this is a rod. The ends of the rod can be rounded, which is sort of what we're seeing here. Let me see if I can blow that up. But they don't have to be rounded. They can be blunt, so that the is like a rectangle, very skinny rectangle, or they could be pointed. And then the third basic shape is a spiral shown here, looks sort of like an S. The spirillum and the spirochetes are the shape. Well, the spirochete is a little more complex, but it is a spiral. Those are the 3 basic shape that most prokaryotes, as well as most bacteria, have. Okay. Now, this is showing you the 3 different types of a spiral, and a vibrio can be considered half of a spiral, or it could be considered a half, not a half, a bent rod. So the vibrio can go either way. You can count it a rod, you can count it as a half of a spiral. Okay. And there are at least one family of vibrio bacteria. Cholera is a member that is a vibrio. Spirals can be the basic shape like this, or they can be multiple spirals, and these we call spirochetes. So they are a spiral, but they're a little different, a little more specialized. There's a little difference that the spiral tends to be rigid and the spirochete is more flexible. It tends to bend more easily. Both the spirals and the spirochete have a unique corkscrew type movement where they move like a corkscrew, opening a bottle of wine when you're trying to put the corkscrew into the cork. Okay. I think I'll show you later. I don't seem to have it on this slide, but maybe in the lab, we'll talk about the movement of the spirochetes, or maybe it's later in this lesson, but I've got a little movie about it. Prokaryotes do have some different shapes, which we call unusual shapes, like there's a star shape, Stella bacteria. This is one family of bacteria, which are this shape, and that's actually an electron microscopic image of 2 Stella bacteria. And then there's actually, it says square shape, but if you look at that, that's more rectangular. The Haley Arcula and Archaea, which are squarish or rectangular. And right there is actually an actual electron microscopic image, and you can see that's clearly a rectangle. Most bacteria are monomorphic, meaning they take on a single shape, like all of these are rectangular, all of these are star-shaped. Where do I have some? All of these are cocci, all of these are bacilli. So those are monomorphic, but there are a few bacterial species which are pleomorphic, and that means they take on many different shapes, such as this species of bacteria right here. Different shapes. That one actually looks somewhat like a rod. I'm sure if I were to look close in here, that's approaching a cocci. It's not quite a cocci. And then these, I don't know what you want to call them. They're just weird shapes. That might look like a vibrio. Okay. But that one, and that one's bent. That one looks like dumbbells. Pleomorphic. Generally, if the bacteria cell lacks a cell wall, and there's only one family of bacteria that lacks a cell wall, the members of that family tend to be pleomorphic in their shape. Let's talk a little bit about the cellular growth arrangement that bacteria can have. The cellular growth arrangement is when the cell divides, how the cells look. You can have, for cocci, you can have singlets, single cocci, which is shown right here. And actually, let me blow this up. Well, those might be singlets. It's hard to tell because they might be hooked there. There's also diplococci, and that one right there is a diplococci. This one right there could be a diplococci. I'm not sure how many are there, but that's clearly 2 right there. And that's when 2 cells divide and then they stay stuck together. They're dividing on one plane, which is diplococci. And if they were to divide again and stay stuck together, we'd have 2 more right here, actually 1 here and 1 there, I guess. And that will eventually form a streptococci, where the cells are all stuck together in a line. Okay. I'm not sure if you only need 2 diplococci stuck together to get a streptococci. I'm not sure what the dividing line between a streptococci is and what one is not. Obviously, a diplococci is when there's only 2. But 3 is not usually considered a streptococci. You can have a tetrad, and that's when the cell divides and then divides again. It forms a tetrad. And they divide essentially on 2 different planes, one going that way and then one going that way. The sarxinia are when 8 cells stick together. And by the way, you can actually see a streptococci here, although it's going over another streptococci. And this one's hard to see right there. We have a tetrad right there. And that's another tetrad right there. Sarcsinia is when we have 8 stuck together and there's just too many of them to see, but that's probably a Sarcsinia there and a Sarcsinia here. But there's another start of one, and probably there's another one there. The Sarcsinia divide on 3 different planes, why they give rise to a Sarcsinia. And then in cocci, we have some which form a cluster like grapes, and this we call staphylococci. Staphylococci literally means a cluster of cocci, or a cluster of ball-shaped cells growing like grapes, staphylococci. And if you look at that, put a stem there or there, that sort of looks like a grape cluster. And there's an actual picture of staphylococci. Oops, too far. Right there. Now, the bacilli also have cellular growth arrangements, but they have fewer than the cocci. First, you can have a singlet, meaning a single bacillus. And that's kind of shown there and there, and I guess there. You can have diplobacilli shown here, and I sure don't see a diplobacilli in here. Maybe this one and that one, but it's hard to tell. And most of the other are not diplobacilli. But that one shows that they're the artist. You can have a streptobacilli, and that's where the bacilli hook together on the long end and form a chain. So right there we have a chain of streptobacilli. And here's probably another chain. You can have the bacilli hooked on the short end, so they're not going to look like a sausage link. They will be looking like this, where they're hooked on together on the long end. And if you flip that 90 degrees, so that's the bottom and this is the top, this looks like a palisade fence. And this is why we call this cellular growth arrangement a palisade, because it looks like a palisade fence. Those of you who read Mark Twain, you remember, I think it was Tom Sawyer, who was painting the palisade fence, and he tried to get Huck Finn into painting it by saying it was really nice and fun painting it or something like that. Palisade fences aren't as common today, but apparently in Mark Twain's day, they were very, very common. All right, any question about any of that? Now, this is showing you not a cellular growth arrangement, so it actually shouldn't be in this slide. It's showing you a different shape of the bacteria. So let's come up here. Is it there? Yeah. And that is, you can shorten the rod and shorten it and shorten it and shorten it. And if it has rounded ends, you will pretty soon be getting a shape close to a coccus, but it will be a little bit longer than a coccus. And that we call a coccobacillus, right there. And this is showing you coccobacilli. That's a little bit longer than a cocci, shorter than a rod. These are 2 cells which are dividing. That's why that looks like a rod there. But if you get to the smaller ones, that's a coccobacillus. And there's clearly a coccobacillus. That one there almost looks like a cocci, but that one's a coccobacillus. And it looks like this is about ready to divide. Any questions about shapes and the cellular growth arrangement? So remember, the cellular growth arrangement is not the shape of the cells. It is how the cells stick together after they divide. when you have a cellular growth arrangement, you never have only one. Like streptobacilli can occasionally break right there, in which case you'll have diplobacilli and streptobacilli in the slide. And then the diplobacilli can occasionally break right there, and then you'll have singlet bacilli, diplobacilli, and streptobacilli in the slide. So the moral of the story is, for the cellular growth arrangement, you will see more than one, with one exception. Okay? If the cell only forms singlets, like a singlet bacilli, then that's the only cellular growth arrangement you'll see. But if it forms like a streptobacilli, you'll see streptobacilli, diplobacilli, and single bacilli. In which case, what you do is you state the highest number, that this cell is growing like streptobacilli. Any question about that? And remember, with cocci, you have more cellular growth arrangements. For the streptobacilli, you'll have diplobacilli and singlets. For the tetrads, you'll have diplococci, excuse me, I didn't say that right. Tetrads, diplococci, and singlets. For the sarxinia, you will have sarxinia, tetrads, diplococci, and singlets. Okay. For the staphylococci, you'll definitely have singlets, probably diplococci, and maybe you will only have staphylococci. You don't usually have the others, although you could have the others. All right, any question about any of that? Is it
On one of the other slides, I saw that it mentioned that there was 2 definitions for bacillus, but it doesn't say what the definitions are.
yes, there's 2 definitions for bacillus. One is a cell shape, what the cell looks like. And the other is there is a genus that is named Bacillus. Okay. Just checking the time here. It's actually 6:04. This clock is not correct. I think I explained that the time on its own changed. And so I got to take it again and hope the time doesn't change. I can't change the time. So I have to take it into the college to get it to change. The point of that is that if it is the family of Bacillus, it will always be capitalized B. If it is a shape of bacteria, it'll be a lowercase b. And that's one way you can tell whether they mean the family of Bacillus or if they mean the shape of Bacillus. You should also not get confused for Streptococcus. There is a family called Streptococcus, and it will have a capital S. And that's very similar, although different, from Streptococci, which should be a lowercase s, and it is the cellular growth arrangement. Okay, so the family is not called Streptococci, it's called Streptococcus, but it's similar to Streptococci. So don't get confused by those two. And yes, the family of Bacillus has the shape of bacilli. And then the family of Streptococcus has Streptococci. And that's why they're named that. All right, let's talk briefly about the structure of a typical prokaryotic cell. Here we're looking at the different typical structures on a prokaryotic cell. You should know that all of the red ones are something that all bacteria have. So all bacteria have a cytoplasm, all bacteria have a ribosome, all bacteria have a plasma membrane, and all bacteria have a nucleoid containing region where we'll find the DNA. It is not a nucleus. There is no division between the nucleoid region and the cytoplasm. Okay. Now, of cells, all cells have 3 of these red ones. All cells have cytoplasm. They do have ribosomes, plasma membrane. But the third thing, instead of a ribosome, is all cells have DNA. So you should know that all cells have cytoplasm, all cells have DNA, all cells have plasma membrane. And then all bacteria have 4 things. They have a nucleoid region. The nucleoid region is just the area of the cytoplasm where the DNA happens to be floating. Remember, the DNA is on 1 chromosome, and wherever it floats, it stains a little differently, like right there. And that region we call the nucleoid region. But there is no distinction between the nucleoid region and the cytoplasm because there's no membrane separating them. The nucleoid region is wherever the DNA moves to in the cytoplasm. All right, let's talk about the glycocalyx, one of the structures of some bacteria, as well as archaea. And actually, there's a few eukaryotes that have a glycocalyx as well. The glycocalyx is secreted by many prokaryotes, but not all. It is outside the cell wall, so we do not consider the glycocalyx to be a part of the cell. The last part of the cell going outwards is the cell wall. So the cell wall is considered a part of the cell. Trying to check the time here. And it is secreted by the cell. The glycocalyx is usually sticky, and there are 3 types. 3 types of glycocalyxes. The one we'll talk about the most in this class is a capsule. The capsule is a neatly organized structure. It has molecules in it besides water, and it helps the cell. Helps protect the cell from phagocytosis. The capsule is something you can see under the microscope, the light microscope. And it's the only one you can see under the light microscope. The slime layer is similar to the capsule, meaning it's secreted by the cell externally, but it's unorganized. And the slime layer is the vast majority of the molecules in the slime layer is water. It's loosely organized. When you look at it under the light microscope, you cannot see it with the light microscope. And that's because it's mostly water, it's transparent. And it does not stain with the capsule stain either under the light microscope. So to see the slime layer, you have to look at the cells with an electron microscope. The third type of a glycocalyx is the extracellular polysaccharide, which is a secretion around the cell that cells make when they're in a biofilm. And this extracellular polysaccharide secreted by the cell is actually what makes up the biofilm. Okay, any questions about that? So this one, the extracellular polysaccharide is a little different. The cell has to be in a biofilm, and the biofilm is actually secreted by the cells to create the biofilm. Any questions about any of that? All right, the flagella is another structure on cells. Some cells, I should mention that the glycocalyx is something that not all prokaryotic cells have. Most eukaryotes do not have a glycocalyx. Okay. But there are some that do. The flagella is a structure found on cells. And if the cell is a bacteria and the cell moves, then that bacteria has flagella, at least one flagella, which would be actually one flagellum. So the flagella is a structure for locomotion. And if the bacteria moves, meaning swims in media, liquid media, then it has at least one flagellum. And flagellum is singular, flagella is plural. The flagella is mostly outside the cell wall. So most of this right there is outside the cell wall. It's a whip-like structure that moves to propel the cell. But there is a little bit of the flagellum or flagella, which is in the cell wall, this region right there. And there's a little bit, the very end of the flagella, which is actually in the plasma membrane. So most of it is outside the cell, but there is a little bit which is in the cell wall and even a smaller amount in the cell membrane. Flagella are mainly made up of the protein flagellin. You don't need to know the basic arrangement of flagella, except for one question on one of the worksheets. It asks you, I think you've already turned it in actually. If you have only one flagellum on one end, we call it monotrichous. If you have two flagella, one on each end, we call it amphitrichous. If you have multiple flagellum, meaning two or more, on one end and the other end, we call it lophotrichous. And then if you have flagella all over the cell, we call it peritrichous. So you don't need to memorize these different terms. I only showed it to you in case you have to know the term to answer one of the worksheets. When you see flagellum on a slide, you will have more than one type, unless it's only monotrichous. The reason being is the peritrichous flagellum, when it first puts out a flagellum, I guess peritrichous flagella, when it first puts out a flagellum, it'll be monotrichous. And then it'll put out a second flagella, and it will be one on each end. So it'll be amphitrichous. And then it'll become lophotrichous, given more time, putting out more flagella. And then lastly, it'll become peritrichous. So most flagellar arrangements, you'll see more than one in the slide. There's only one exception, and that is if the bacteria are monotrichous, you'll only see monotrichous. Although in this case, you could see some that have not put out a flagellum yet, but most would be monotrichous. All right, flagella allow cells to move. It's the only way that bacteria cells, when they're swimming, can move. The cell will rotate the flagella to either run or tumble. And this allows the cell to have taxis. Taxis is the movement of a cell toward or away from a stimuli. Like these cells here are moving that way, and the light is this way, so they are moving away from the light. And these are obviously not photosynthetic cells because they'd move towards the light. Taxis is important for cells because they allow the cell to move to a better environment. We can have two different types of taxis. There's chemotaxis, which the cell responds to a chemical. And then there's phototaxis, where the cell responds to light. As I stated, most photosynthetic bacteria will move up towards the light. All right, I think we'll go ahead and talk about the axial filaments. They're also called endoflagella. I tend to use the term endoflagella. The axial filaments are a special flagella, or flagellum, I guess I should say, because there's only one, that is inside the cell wall of a spirochete. So endoflagella are only found in spirochetes, and all spirochetes have an endoflagella. The endoflagella does allow the cell to move, but this movement is in a corkscrew-like movement. And here we're looking at
As I'm in the midst of applying for residency right now, wish me luck.
turn that off while going through the commercial. Come back to here and say that here we have the cell. Let me blow this up. Come on, blow up. The endoflagella wraps around the spirochete, and when it moves, you'll note that, oops, a little too far. Let's go this way. There it is. Here we have the cell wall in light pink or light purple, and the axial filament or endoflagella is inside the cell wall. Try and let it be in further. Scroll it down. All right, so the endoflagella is inside the cell wall. And let's look at the movement now. There we go. Endoflagella, a corkscrew-like movement. Let me see if I can blow that up. And there you can see the spirochete is so flexible, it can even change its movement. So it was going up and down, and it can change going up and down just by which way it's having its spiral-like movement. So it moves this way to go that way, and it'll move the other way if it wanted to go up. All right, any questions about spirochetes? If not, I think I'll end here, and I will see you at 6:30 for the lab. All right, bye.